I welcome all the participants in this Science Dialogue series. As you all know, this is free Science Dialogue series, free webinar series. And we are organizing this free webinar series since 2023. I request all of you to interact with a speaker as maximum as possible. And in this series, we have invited today Dr. Som Dev. He did his PhD from a special center for molecular medicine and then postdoc in different institutions, John Hopkins and other institutions. Nowadays, he is assistant professor at Department of Biochemistry, AIIMS, All India Institute of Medical Science, Kalyani. And he has published several very high impacted articles in PNAS and Science and Blood. So that journals are of very high impact. So I welcome you, Dr. Som Dev, and we are grateful that you have accepted our invitation to deliver lecture, because this is also very a good thing to disseminate knowledge, and this is daring thing to disseminate knowledge openly if you are not knowing the standard of the participants. So this is like a national webinar or international. Sometimes participants are from international, different country also. I request all the participants to attend complete lecture. Then I will share you feedback link also in the chat box. If you will fill that feedback form, then you will receive e-certificate. So don't ask me e-certificate since you have joined for 5 minutes or 10 minutes. You have to fill that feedback form, and that will be shared after lecture in this chat box. Same time during discussion. So thank you again, and I apologize for delay, technical things, due to technical things. Now platform is yours, Dr. Som Dev.
Good evening, everyone. First of all, I would like to thank Dr. Gopal for the invitation and giving me opportunity to share some of my work here with students and young researchers. So topic for today is sterol misbalance contributes to pathogenesis of Wilson disease. Wilson disease, it's a potentially fatal metabolic disorder caused of copper metabolism, and it is caused by mutation of one of the copper transporter gene named ATP7B. Sterols are a type of lipid molecules, and in this, these are multi-ring structures. Cholesterol is one of the example, one of the most common example. And here in this project, what we have found that the downstream of cholesterol comes the oxidized form, that is oxysterols, and the sulfated oxysterols. The balance of those oxysterols and sulfated forms that dysregulates nuclear receptor functions, and that ultimately leads to Wilson disease pathogenesis. Next slide. So this is, can you go back? Please go to previous slide. No. Previous, back. Previous one. Previous. Okay. So as you can see here, the structure of cholesterol and oxysterols, there is only the oxidation of cholesterol that leads to oxysterol synthesis. Please, next slide. So I have nothing to declare. Next. So in the next 30 to 45 minutes, I will try to bring you attention regarding what is the importance of copper, how body regulates the copper homeostasis, what are the management challenges of Wilson disease, and briefly about pathogenesis of Wilson disease. And in a little more detail, we will discuss the molecular mechanism, how this copper overload disorder that leads to sterol misbalance, which in turn is a driver for nuclear receptor dysfunction in mouse model of Wilson disease. Next. So copper is very essential for life, especially in higher eukaryotes. The organism can't survive without copper redox activity. And this redox activity, liver is the major organ of copper homeostasis. And mutation in ATP7B causes copper accumulation in tissues, especially in liver and later on in brain, and also leads to metabolic abnormalities, gradual loss of liver function, and if not treated, it leads to death. As you can see from the pictures on the bottom, on the left side is the liver disease scar liver when you see macroscopically, and on the right bottom panel is the staining done for the copper in the liver sections. So to understanding copper metabolism is very important to understand the pathogenesis of Wilson disease. Next, please. So this Wilson disease, it's a rare genetic disorder, autosomal recessive nature. And this was named after this doctor, physician scientist, Samuel Wilson in 1912. He basically presented a case. Basically, he presented a case, and it was this gold medal awarded MD thesis, which he showed a case of the patient of familial history of neurological symptoms with liver cirrhosis, and which ultimately lead to the name coined after him, the Wilson disease. So the global prevalence is 1 in 30,000. Next slide. So, but in India, the estimated prevalence of Wilson disease in India is higher, lot higher. In recent publication in this year only, in multicentric pan-India study regarding Wilson disease, this prevalence is around 1 in 18,000 only. And approximately 50% of the cases in India have positive family history and consanguinity. That means blood relation. So this is due to intra-ethnic, intra-community marriages, in simply put, cousin marriages. So that is leading to 50% of homozygosity in Wilson disease. And this gene ATP7B, it's a big gene, which has more than 750 mutations so far reported. Next. So what about the liver disease progression? So liver diseases from the healthy liver, first there is so on the liver disease progression, it starts with fat buildup. That is, the state is called steatosis of fatty liver, which then undergoes inflammation and fibrotic changes. There is a liver fibrosis happens, and all these three stages up to liver fibrosis, these are reversible stages. These can be prevented. But once there is a permanent scar formation or cirrhotic stage, that's the irreversible stage. And the liver is basically completely damaged, and which can't be reversed. And the common causes of liver disease are alcoholics, the alcoholic liver disease, metabolic acidotic steatotic liver disease. The most common causes are type 2 diabetes, obesity, viral hepatitis, and the genetic causes are the most common are Wilson disease. So it is important to understand that all of these moieties, the causality, it leads to ultimately liver disease damage, which goes through steatosis, fibrosis, and ultimately cirrhosis. And ultimately, after cirrhosis, the many patients then end up into hepatocellular carcinoma. Next. So to understand the copper homeostasis in the body or the overall systemic copper homeostasis, copper is absorbed in the intestine. It's reduced from cupric to cuprosine, which enters the enterocyte through this copper importer CTR1, and then exit out through another exporter ATP7A into the portal circulation. Through portal circulation, copper reaches the liver. Within the liver, again, the copper enters the hepatocytes through this importer CTR1, and within the cytoplasm, copper is utilized for various biological function, like in mitochondria, in energy metabolism, and it also goes to superoxide dismutase 1. And the majority of the copper there is handled by this ATP7B copper transporter present in the trans-Golgi network. So within the liver, it performs dual function. It delivers the copper to the ceruloplasmin. Ceruloplasmin is the major copper-carrying protein in the circulation, which delivers copper to the other tissues. And excess copper is also exported out or excreted into bile or feces through membrane-located ATP7B. And next, please. So when there is a mutation in this ATP7B, that leads to copper overload or Wilson disease and resulting into copper accumulation in the liver. Next. So regarding the current management challenges of Wilson disease, the current management is handled by copper chelation therapy, zinc salts, and if the liver is completely damaged, the transplant. But right now, the copper chelation therapy is the standard of care therapy. But however, all these treatment modalities have limitations like side effects, poor compliance, high cost, potential for neurological decompensation. So understanding copper-induced Wilson disease pathogenesis may lead to alternative or supplemental approaches to current chelation therapies. Next. So as we know that it's well known that high copper and oxidative stress has been reported in Wilson disease patients. There are several reports regarding this because copper is a redox active metal that causes ROS generation. But however, how this copper-induced oxidative stress causes tissue damage at the molecular levels, the details are very limited. So it is important to understand how we can address this tissue damage at the molecular level, how this copper-induced stress is causing this. So that was our idea behind this project. Next slide, please. So the one important thing to mention here is that in Wilson disease or in the mouse model of Wilson disease, ATP7B knockout mice, before the pathological onset, before the liver changes like fibrotic or pathological changes, the two things happens. The copper accumulation build up and nuclear receptor function gets declined. And nucleoreceptors are basically the ligand-dependent transcription factors, which when it goes to the nucleus, it binds to the target genes and regulates their expression. And this high copper-induced stress, in this study, what we have done, we have used this ATP7B knockout mice with established liver disease. Basically, by the age of 20 weeks in mice, we have a full-blown liver disease in these knockout mice, Wilson disease model, and it has copper overload, stress. And here we found out that how this copper-induced stress, which is sterol misbalance, is driving the nuclear receptor dysfunction. Next, please. So now coming to the, first of all, as we discussed again and again that copper induces stress, the most important factor to address the oxidative stress is ANRF2. This is oxidative stress sensitive transcription factor, and it is also called as a master regulator of oxidative stress. So first of all, what we have done, we performed immunofluorescence staining in the knockout liver section. As you can see in the panel A, the knockout liver section has more NRF2 abundance as compared to the age-matched HAT control, and DAPI was used as a nuclear marker. So the first picture or the imaging immunofluorescence suggests that there is more NRF2 abundance in the knockout liver section as compared to the control. And this we also verified by performing Western blot analysis from the knockout liver homogenates, both from the cytosolic fraction as well as from the nuclear fraction. As you can see that NRF2 expression is more in the knockouts as compared to HAT liver homogenates. So both these data suggest that, of course, the NRF2 expression is high in the mouse model of Wilson disease liver, and this is also verified by immunoblotting. Next, please. So we also confirm this increased NRF2 in Wilson disease mice by using the Wilson disease patient's liver sections. As you can see, this is the patient data from the Wilson disease liver sections. As compared to control, the NRF2 expression is higher in both the patients Wilson disease 1 and 2 liver patient liver sections by immunohistochemistry. As you can see from lower to higher magnification, there is more expression of NRF2 in the patient samples as compared to control liver section. Also, there is an interesting part you can appreciate in the 100x. In the second patient, Wilson disease 2, there is a PET cells also. That means this the patient 2 has steatosis also, along with the copper overload problem. You can appreciate there is a big fat cells at the higher magnification in the second patient. So both the data from the mouse data as well as from the Wilson disease patient data suggests that NRF2, which is the master regulator of stress, is high in mouse and the patient settings. Next slide. So how this NRF2 works? Basically, it acts by moving from the cytosol to the nucleus. Then it binds to this ARE element of the target gene. ARE stands for antioxidant response element, which is present in all of these target genes, which belongs to glutathione system, like glutathione peroxidase, and then other synthetase, thyrodoxine system genes, ROS and phase 2 detoxification enzymes, NADPH regeneration, iron metabolism gene, like for example hemoxygenase, ferritin, and NRF2 itself along with its partner MAPS. So all of these are NRF2 target genes. For this, what we have done, we have used this mice liver and sent for RNA sequencing. And this is the heat map data for all of these NRF2 target genes. As you can see that more red color intensity means there is upregulation of most of these antioxidant and cytoprotective NRF2 target genes, which belongs to glutathione, thyrodoxine system, and other genes. Among these upregulated NRF2 targets, the most abundant fold change was for sulfotransferase 1E1. SULT 1E1, it's an important enzyme which causes sulfation of the oxysterols. And next slide, please. To confirm this fold change, we performed immunohistochemistry for SULT 1E1. As you can see, that entire liver section of the knockout, this brown positive staining of SULT1 is expressed throughout the liver section. And same we have confirmed and verified in the patient Wilson disease liver sections that the SULT1E1 expression, it's not just changed at the RNA level, it is also high, as you can see from the immunohistochemistry data. And if there is more SULT1E1, it will lead to more sulfation of oxysterols. So, and the sulfated oxysterols, they are known to inhibit liver X receptor activity. Next slide, please. Next slide. Next slide, please. Shash. Slides are not moving.
Saswat.
Meanwhile, a question पूछ लूं.
Yes, yes.
Please,
Please go
please
ahead.
ask Sipra madam.
Like copper, we use copper pots, right? So that will increase like copper quantity in our blood. So
Right.
how is it helpful or it's going to cause some?
No, it will cause some problem if you exclusively use copper utensil for
Saswat
cooking.
share karo.
So there was a disease called Indian childhood cirrhosis. In 50s 60s, so there was so many patients which were having copper overload in the kids. So the term was called Indian childhood cirrhosis. That was due to exclusively using copper pots or copper utensil for cooking purpose. So the
Okay.
excess copper was coming. But nowadays nobody use, like very rarely we use copper utensil for cooking. So this problem of Indian childhood cirrhosis is nowadays it's sort of rare, but it does happen that if you exclusively use copper pots, there will be absorption of copper more into the gut.
Saswat.
So that means it's at all we should avoid.
Yeah, so the thing is we should avoid. So minimal, I don't think it will create a trouble. If it is exclusively using copper pots, then it will be a problem, especially for the children.
Yeah, for drinking water, like many Vaidyas also suggest that it makes water alkaline.
But that is not good, na?
Yeah, it's not.
So it's contradictory to existing this, Hannah.
No, the thing is that it is coming into the system. It will build up because the system is trying to handle it. But if the threshold is crossed, it will be
Okay.
overburdened like Wilson disease.
Mm.
So, so Shashwat, can you make it hide this bottom thing
Yes, yes.
and move to the next slide? Okay, so far, so what we have understood that this copper overload induced stress, there is increased NRF2 expression, not just in the mouse model, but also in the patient liver sections. So that tells us that, and also apart from that, the genes which are dependent on NRF2 or NRF2 target dependent genes, all of these cytoprotective and antioxidant genes, all of them were upregulated in the mouse liver, Wilson disease mouse liver. And then among them, the sulfotransferase 1E1 was the major one, which was significantly induced, more than 5-fold change. That we also now shown and confirmed, verified by immunohistochemistry data from the patient settings. And now we try to dig out this sulfotransferase 1, how it works, where it comes in this cholesterol metabolism. So cholesterol, as you see in this figure A, it gets oxidized and hydroxylated into oxysterols, which are natural ligands for LXR-RXR activation pathway. And whereas the oxysterols, they undergo sulfation with the help of sulfotransferases that will lead to synthesis of oxysterol sulfates, they are antagonists of LXR-RXR activation pathway. And cholesterol can also directly sulfate it into cholesterol sulfate that will also inhibit LXR-RXR pathway. So we thought we will use our bulk RNA sequencing data and check out all of the genes or enzymes which causes this hydroxylation and sulfation. And first we checked all the CYPs which causes oxysterol synthesis. As you can see in panel B, all of these enzymes were relatively lower in the knockouts as compared to the control control mice sample. They were decreased. Interestingly, the downstream enzymes which causes sulfation, all of them were upregulated, like 2 to 500-fold, except one salt, that is SALT28, which is known to cause sulfation of bile acids. So this opposite changes in the enzyme which causes oxysterol synthesis and oxysterol sulfate synthesis, that will net result will be inhibition of LXR-RXR activation. We also verified this increased SALT1E1 transcript by qPCR. As you can see in the panel D, that this SALT1E1 expression by real-time PCR, it was more than 100-fold at 12 weeks. And it with the liver disease is progressing at 20 weeks, the SALT1E1 expression was almost 200-fold as compared to the control heads at the same age group at 20 weeks. So this tells us that SALT1E1 is getting induced in the liver cells, and this will suggest that net result will be inhibition of LXR-RXR activity. Next slide, please. So to verify the changes which are happening, opposite changes happening in the oxysterol enzymes and the sulfated enzyme, what about the oxysterol level itself? So then what we have done, we performed LC-MS and checked the oxysterol content. As you can see in the top panel, starting from cholesterol onwards, all the oxysterols, whether it is 4-hydroxycholesterol, 7, 24, 25, all of them, their content was low in the knockout liver as compared to the head control. Whereas the downstream sulfated forms, especially 24 oxysterol sulfate, 25, 27 DHEAs, all of these sulfated sterols, they were significantly induced in the knockout liver. So this tells us that overall from both these slides, it suggests that the decrease in the natural activator of LXR or endogenous LXR agonist and increase in expression of LXR antagonist, that is sulfated oxysterol sulfates, will lead to persistent inactivation of LXR activity. Next slide, please. So to address that, what we have done then, we thought, okay, SALT1E1 is high. There is opposite changes in the enzyme which causes oxysterol and sulfated sterol synthesis, as well as their levels or contents. So we thought, if we block this enzyme SALT1E1, what will happen to the oxysterol levels or sulfated sterol level, whether it will improve LXR activity or not. So what we have done here, we have used 12 weeks old ATP7B mice and injected 3 times with triclosan. It is a known SALT1E1 inhibitor, or with the corn oil as a vehicle, as a control in the mice. As you can see in the panel B, the untreated chaos with corn oil has low oxysterols, whether it is 24, 25, 4, 7, all of them are low. But treatment with triclosan for 3 days, it led to significant improvement in especially 24 oxysterol as well as 25 oxysterol with triclosan as compared to corn oil treated knockouts, which is shown in the green bracket. And what about the sulfated moiety in the panel C? You can see 24, 25, DHES, all of these sulfated form of oxysterols, they are reduced. So the activator of LXR, that is oxysterol 24, 25 oxysterols, they are improved by blocking SALT1E1, and the sulfated form by blocking SALT1E1, they were relatively reduced. So the net result will be increase in LXR activity. So what we have done, we checked LXR, RXR target genes. So LXR, RXR, what it does, basically normally it is a anti-inflammatory as well as it induces lipid metabolism and it inhibits inflammation. But in the untreated KOs where there is less LXR activities, as you can see, all these inflammatory markers starting from inducible nitric oxide synthase, cyclooxygenase 2, IL-6, TNF alpha, IL-1 beta were high, which was like 3 to 12, 15 fold, all of them. And treatment with this SALT1E1 inhibitor tricosan, all of these inflammatory markers, they are reduced, they come closer to the control, the blue hat level. Whereas the lipogenesis gene, fatty acid synthase, SED1, their expression was improved with SALT1E1 inhibitor. So all of these data suggests that if we block pharmacologically SALT1E1 enzyme, it reverses partially the sterol misbalance as well as it also improves LXR activity. Next slide. So next we thought, okay, why not use the known LXR agonist and see what will happen to inflammation and fibrosis, whether it will also change instead of using the SALT1E1 inhibitor. So first we thought, let's check the copper levels, which we know that it is very high in the knockout mice. So treatment with LXR agonist, this is T0901317. It is a known LXR alpha beta inhibitor. So treatment with LXR agonist, it doesn't impact much the copper levels. Irrespective of the genotype, male or female, the copper levels are still very high. There is a minute, minimal decrease in copper levels as compared to untreated KOs when we compare with KO plus T0, that is basically LXR agonist. But overall copper levels were very high. There was not much significant change irrespective of LXR agonist treatment. So then we thought, let's check the liver function test. So we checked serum total bilirubin. So liver functions in the untreated KOs was high in the mouse data. And the treatment with LXR agonist, the total serum bilirubin was coming back to the hat levels, the control level. And the serum albumin, which was in the untreated knockout mice or the Wilson disease mice, the serum albumin content goes down. It was significantly low, which is shown in the red symbols. And treatment with agonist in the right side, there is improvement in the serum albumin content. So that means when you are treating with LXR agonist, by increasing LXR activity, there is improvement in liver function, even though the copper overload is still intact or still there. Next slide, please. So next we thought, what about the histopathology? So as you can see in the H&D staining, the KOs, the Wilson disease mouse knockout liver has enlarged hepatocytes. There is karyomegaly. There is also immune cell infiltration and vacuolation. But when you treat the fourth panel, the knockout with the LXR agonist, all of these inflammatory and histopathological changes are minimized. And also what we have done, data is not shown here. We scored all of this slide by blind score by pathologist. And even there was almost 40 to 50% reduction in the inflammatory score, which was given for the untreated KOs. There was a reduction in the inflammatory score by 40% in KO plus LXR agonist treatment. So that means it is also not just improving the liver histology, reducing the pathology, also reducing the inflammation by treating with LXR agonist. So next slide, please. So, since the inflammatory cytokines or inflammation that leads to activation of hepatic stellate cells, that is the prerequisite for liver fibrosis to happen. And there is also the induction of collagen 1 and TIMP1 fibrotic markers. As you can see, the untreated knockout mice, there is more than five-fold induction in collagen 1 alpha expression, mRNA expression. So is the case with TIMP1 marker. But treatment with LXR agonist is T0. So both of these fibrotic markers comes back to the HAT levels. So all these data suggest that this, if we are using directly LXR agonist, despite having copper overload, it does improves liver functions and also reduces liver fibrosis. Next slide, please. So next we thought, what is the pathway or the signaling mechanism of this fibrosis? So using our bulk RNA sequencing dataset, we already figured out by pathways enrichment analysis that TGA beta was one of the most enriched upstream regulator, and its expression was also high at mRNA level or RNA level. So we thought, if we do immunohistochemistry, what will happen? Whether there will be really show the change which we observed in the RNA sequencing data. Yes, indeed, as you can see in the 100x knockout section, for TGA beta, the signal, there is a brown TGA beta immunosignal, both in the hepatocytes, the bigger cells, as compared the smaller non-parenchymal liver cells, indicated by red arrow. So that means there is increased in the TGA beta ligand, and treatment with agonist, LXR agonist, there was a reduction in TGA beta signal. Next slide, please. So this TGA beta signal, as I mentioned that in RNA-seq data, TGA beta was high at RNA level. Now we verified it by immunohistochemistry. So was the case to which it binds at the membrane, TGA beta receptor 2. Its expression was high at the RNA level in using sequencing data. We verified by immunofluorescence data. Here, what we have performed co-immunostaining with desmin. Desmin is a known marker of activated stellate cell, or you can say simply the fibrotic marker. Its expression is high in the knockout mice, which is shown in green colors. And TGA beta receptor 2 expression was also high in the knockouts. And the both of these induced markers, stellate cell marker desmin and TGA beta receptor 2, they were co-localizing at several places in the liver section. And the treatment with this agonist, there was a reduction in the expression of fibrosis as well as the TGA beta receptor 2. Next, we thought that if, okay, TGA beta is high, TGA beta receptor 2 to which it binds, it is high. What about its functionality? Whether this change in at RNA level or at the immunohistochemistry or immunofluorescence level, whether it will show in the functional change also. What about the downstream signaling which gets initiated by TGA beta receptor 2 complex? So then we check the TGA beta signaling in further. Next slide, please. So this TGA beta signaling happens, as you can see here, the TGA beta ligand binds to receptor 1 and 2, and then it can go through the classical SMAD signaling pathway. First, there will be a binding of SMAD2, 3, there will be phosphorylation, then will come SMAD4, which will bind to SMAD2, 3 complex, which will move to the nucleus and trigger the gene expression. Or TGA beta signaling can happen through non-canonical TGA beta signaling pathway. It is also called MAP kinases signaling pathway, which happens through this ARC signaling pathway, P38, JNK kinase pathway. And the right side panel, you can see that when we performed the Western blot analysis, all of this, not just the TGA beta, TGA receptor 2, what I just discussed in previous slide, also the these kinases or enzymes which belongs to the classical SMAD and non-canonical MAP kinases pathway. So all of them, their protein levels were high in the untreated KOs. But interestingly, the treatment with this T0 LXR agonist, SMAD2 levels remained high. They were still 1.5 to 2-fold high at protein level, whereas the treatment with LXR agonist, it reverses the changes in ERK1, 2, P38 alpha beta, and JNK. So that tells us that the treatment with LXR agonist It reduces fibrosis in Wilson disease mice, but primarily it is possibly happening through non-canonical TGA beta signaling pathway, that is MAP kinase signaling pathway. Next slide, please. So in summary, copper-induced oxidative stress, it triggers NRF2-mediated stimulation of sulfotransferases with resultant increased synthesis of sulfated sterols, and decreased synthesis of oxysterols creates a sterol misbalance. This sterol misbalance helps to explain the link between oxidative stress and dysregulation of nucleoreceptor genetic pathways, resulting in fibroinflammatory liver disease. And our preclinical data with LXR agonists demonstrate that it can partially improve liver inflammation and liver function, as well as fibrosis in Wilson disease mice with established liver disease. Next slide, please. Next slide. Shashwat? Okay. Okay, so
Okay.
so in this, basically in conclusion, slide mode.
No. विंडो किया है कि नहीं?
Okay. So in conclusion, what I have discussed in last half an hour, we have the normal condition, or in the normal wild type mice, we have a functional ATP7B that maintains the copper homeostasis in the liver. We have a normal regulation of cholesterol metabolism through the CYPs and the SALTs. There is a balance of LXR, RXR activation. So there is a normal lipid metabolism, no inflammation and fibrosis in normal settings. Next slide, please. So whereas in Wilson disease mice, first there is a mutation, inactivation of the ATP7B causes of copper overload. That leads to increased oxidative stress, resulting in increased expression of sulfotransferases and decreased expression of CYPs, and that results in inhibition of LXR, RXR activation, resulting in a reduction in the lipid metabolism and upregulation of inflammation as well as fibrosis, as you can see by these markers for inflammation and fibrotic markers, resulting in inflammation and fibrosis. When we treat with this or LXR agonist, which is shown in green bar, T0, it improves the LXR RXR activity, resulting in the upregulation of lipogenesis and reduction in inflammation and fibrosis. The reduction in fibrosis is possibly by the non-canonical TGA beta signaling pathway. Next slide, please. So the take-home message is that the sterol misbalance is an important driver of Wilson disease pathology by nucleoreceptor pathways downstream of elevated copper. And LXR agonist could be tested or explored to supplement copper chelation therapy in Wilson disease. Next slide. So if anybody wants to read this, whatever we have discussed in last half an hour, you guys can read this article. It's quite a big article, but you will get a lot of information regarding the Wilson disease pathogenesis and how there is a sterol misbalance is happening. Next slide. Also, what we have noticed recently in another publication in liver fibrosis, that this liver fibrogenesis, there is a changes in the microtubule stability also. There is a microtubules destabilized, which is concomitant with liver fibrogenesis, that is also leading to overall Wilson disease progression. So this was published recently. Next slide, please. So lastly, I would like to acknowledge my the mentors, gastroenterologist and the physiologist from the Johns Hopkins, Professor Svetlana and Dr. James Hamilton, and the funding from the NIH. And here at AIIMS Kalyani, I'm looking forward to understand the Wilson disease and other mental disorders like thalassemia and anemia in greater detail. So I'm looking forward if anybody is interested to work with the metals, which are very essential for life. Next slide. Thank you for your attention.
थैंक यू वेरी मच. अल्टीमेटली टेक्निकल फॉल्ट आया बट बहुत अच्छा रहा डिजीज से रिलेटेड. तो मतलब कॉपर वाले कंटेनर में नहीं पीना चाहिए, है ना? यही
पीना चाहिए,
कंक्लूजन है.
बट ज्यादा कुकिंग का थोड़ा देखना पड़ेगा.
कुकिंग का?
सो कुकिंग इन कॉपर यूटेंसिल्स, कॉपर पॉट्स.
अच्छा, कुकिंग नहीं करना है ना?
Extensive. Sometime cooking, it will not make much
एक्सटेंसिव
difference.
मीन्स हाउ मच डू यू सजेस्ट? हाउ मच माइक्रोग्राम, पिकोग्राम, नैनोग्राम पर डे अगर होगा तो एक्युमुलेट होगा? ये यानी कंसंट्रेशन जो
No, I think the data which was observed for this Indian childhood cirrhosis, which was a known condition in 50s, 60s, that means they were consistently using copper pots for cooking.
Okay.
Occasional cooking, it will not make much impact for the adults. Especially the kids or the children are more vulnerable because they do not have the system which is properly developed. Like some of these, like even the ceruloplasmin, which carries copper in the circulation. In the newborns or kids, their levels are less expressed in the. It takes them a few years, 2-3 years, to have the adult level of ceruloplasmin expression.
ऐसा नहीं डाटा है कि हाउ मच माइक्रोग्राम विल एक्युमुलेट कॉपर
So
एंड
that I
देन
have to check, because nowadays these conditions are more or less obsolete, especially this copper pot condition.
अभी भी लोग जो है भोज में या कॉपर का लोटा कॉपर का ही होता है ना, जो
No, I
सुबह
think
सुबह
that will be minimal. If you are using cooking mein you have to
लीचिंग ज्यादा होता है.
that will be more leaching and extraction of copper into the your food.
ओके ओके ओके. बट वी शुड स्टडी है ना हाउ मच मिनिमम कैन बी
But this
टॉलरेटेड. और
problem as such, we are getting patient, even the Wilson disease patient. This is covering almost 7 to 18% the child with pediatric liver problem. This is comprising almost 18 to almost 20%, only Wilson disease.
ओके
So in northern India, it is like 8%, 9% is comprising pediatric liver problem. Wilson disease is covering 7 to 10%. So we do get patients. Another problem with the Wilson disease, because it's a genetic problem, the cousin marriages. Simply putting, cousin marriages. Avoid marriages within intra-ethnic, intra-community groups. Please avoid marriages within the close relatives.
बाहर करना चाहिए, है ना?
Yes.
अपने कास्ट क्रीड के बाहर करना चाहिए. वो तो बहुत पहले से है. एनी क्वेश्चन फ्रॉम पार्टिसिपेंट्स? आई हैव शेयर्ड Google form, feedback form. So those who need e-certificate, fill that Google form, and don't remind me every day that when you will get certificate. We will send after accumulating 2-3 lecture, we send, है ना? So just fill that. And I have also shared several social sites, my LinkedIn, YouTube, and other things. So you should join if you are new to this Science Dialogue series, and we continuously conduct. So thank you very much.
Thank you,
Dr. Som Dev, for joining us.
I apologize for all these technical glitches.
बहुत technical रहेगा थोड़ा. Any person have any question, any complimentation, any suggestion regarding talk or regarding Science Dialogue series? You can suggest. You can join WhatsApp group also. कुछ suggestions हैं किसी के? नहीं. So thank you. Thank you very much. We are grateful.
Thank you.
And we will again listen you in future on another topic.
Thank you.
ठीक है ना? Thank you सौम्यजीत and शाश्वत. थोड़ा इंटरनेट कनेक्शन थोड़ा अच्छा रखना चाहिए था.